Hello everyone, I'm Sheena and welcome to my channel Comprehend Biotech. The topic for discussion today is RNA-seq or RNA sequencing. This session is specifically designed for undergrad students to understand the basics of RNA sequencing. It's kind of like an introductory class. Now, RNA sequencing has four major steps. Isolation and selection of RNA species, library preparation, next generation sequencing, and data analysis. Now, covering all of these topics in one single video would take me like more than 2 hours. So to overcome that problem, I have divided this session into 4 parts. Part 1, Part 2, Part 3, and Part 4 that you see on the screen. So all these topics will be discussed over here. In case you're wondering what to expect in each part of the video, here you go. So in part 1, we'll be basically discussing what is RNA sequencing, what is differential gene expression, about RNA isolation and selection, about RNA integrity number, RNA enrichment, library preparation. We'll begin with library preparation, the second step, and we'll look at RNA fragmentation and cDNA synthesis. So these topics will be covered in the first part. Let's see what will be in the second part. Second part basically will focus on the second step, that is library preparation. Under that, we'll first just see what is the basic PCR reaction. Then we'll go into how adapters are ligated, what are Y adapters, what are the basic elements in adapters generally used for sequencing. Then we'll also be looking at bridge PCR or cluster amplification. In part 3 of this video, we'll be focusing on the sequencing step. So we'll be understanding what is next generation sequencing, and specifically we'll be learning what is sequencing by synthesis by Illumina. And under that, we'll also be learning what is paired end sequencing. Okay. So part 4 of the video will focus on data analysis. Under that, we'll be looking at data processing, transcriptome assembly, analyzing different outcomes. Then we'll have a summary of the whole RNA-seq procedure, and we'll be having a little peek into what is direct RNA-seq and single-cell RNA-seq. So all these topics will be covered in these 4 parts of this video. So today we are discussing part 1 of the session, and yes, welcome to the class. Welcome again. The topic for discussion today is RNA-seq, and we are discussing part 1 of this session. So in this part 1, as already mentioned, we'll be discussing the following topics. And in case you already know a lot about these particular topics, you can always check the description of this video. I have put timestamps for each topic so that you can go and understand, or you can go and learn the topics that you wish to learn. So just check out the description, just go click whatever you want to learn. Okay, so let's begin. So what is RNA-seq? So RNA-seq, as the name suggests, it's a sequencing method for sequencing RNA. That means we are going to find out the order of nucleotides in a particular RNA sequence. Okay, so it's a next generation high throughput RNA sequencing method. It's also a quantification method, which is used in the study of transcriptomics, differential gene expression, transcriptional profiling, and single nucleotide polymorphism identification. Okay. It can tell us, that is RNA-seq can tell us what or which genes are activated, okay, what their level of expression is, and at what times they are activated or shut off. We'll be going into the detail. Do not worry. So before we actually get into RNA-seq, a small concept, that is differential gene expression, has to be cleared. Okay, so let me just try to put it in the simplest way possible. Now, if you look at your own body, right, we are made up of different types of cells. Just for an example, we have blood cells in us, we have skin cells, we have nerve cells, right? If you look inside the cells, if you observe the DNA is actually the same in all these cells, whether it is your skin cell or blood cell, the DNA is same. Then what is that difference, or where is the difference arising that makes a particular cell act like a skin cell, another particular cell maybe act like a nerve cell? So that is where the concept of differential gene expression comes. That means if you look at our DNA, we have certain portions in DNA which are called genes. Genes are the actual sequences which code for proteins, right? If you remember the central dogma of molecular biology, you have DNA. DNA is transcribed to mRNA, which is translated to proteins. So expression, in a sense, means which gene is expressing which protein. The production of a protein can be, in very simple terms, be denoted as expression. So differential gene expression would be in a particular cell at a particular time, not all genes are functional. Some of the genes are switched on, some are always off. And which particular gene is on or off will determine which protein is synthesized or how will the gene expression be. In very simple terms, at the birth of a new cell itself, it is predetermined that this particular cell will have a certain set of genes which will be activated and others will be turned off. So that is in a nutshell what is differential gene expression. So this concept was developed and 3 postulates were given to define what differential gene expression is in the early 1960s. So let us see what are the 3 postulates. Okay. So first one is DNA is identical in all the cells that we know. In a particular organism, DNA is identical. Okay. Second one is unused genes are not destroyed, but they retain the potential. Okay, so a gene which is not expressed will not be destroyed off. It will be present there and it can always retain its potential. Okay. Third one is only a small portion of genome is expressed, which is specific to each cell. Okay, that is what we discussed, right? If you have your DNA, all the genes are present in all the cells, but only very less portion is expressed. And that is actually predetermined. Okay, it is specific to specific cells. So these postulates actually help to define what differential gene expression is. So differential gene expression can be studied between different organisms. It can be studied between different tissue samples. It can be studied between different set of samples from the same organism also. So you can do a variety of studies on differential gene expression. Okay, to understand the concept further, I have drawn a simple diagram. Let's just try to understand with that. Okay, suppose you have your normal cells and the same normal cells, we have taken another set which have been infected or mutated. So if you look at the DNA level, the DNA will be the same for both the cells, right? So suppose this portion codes for gene A, this for gene B, this for gene C. Same way over here, gene A, gene B, gene C. Okay, after transcription, how high the expression is or how low the expression is, depending on that, your DNA will be transcribed. Particular regions will be transcribed and you will get mRNA according to it. For example, in this normal cells, gene A is expressed so many times. So you have so much of mRNA, right? Gene B maybe only 1 time, gene C only 3 times. Okay. If you look at the infected cells, the same gene A has been underexpressed, whereas B has been overexpressed and C has been is remaining the same almost. Okay, so all this is happening inside the cell. What if you can quantify this particular phenomenon? What is the change between gene A, gene B, and gene C happening between these 2 cells? What if you can quantify that? To quantify that, you will have to take it out of the cell. So these 2 steps happen inside your cell. The next analysis part, you need to carry it outside the cell. So that is where you'll be isolating the RNA, specifically messenger RNA or mRNA, which is the one that codes for proteins. Okay, and then you will analyze it. You can use different techniques, for example, RNA-seq, which we are going to study today, and SAGE and other transcriptional profiling methods. Okay. SAGE, I have already discussed, or serial analysis of gene expression, I have already discussed in a previous video. If you have no clue about it, please go and check that out. It's a very interesting video of how transcriptional profiling can be done using SAGE technique. Okay, please do check that out. So after this analysis, we might be able to find which gene is expressed or underexpressed, overexpressed or underexpressed. When is it done during a cell's life cycle? When is it underexpressed or overexpressed? And how much is the expression level? So all those things can be understood by carrying out gene expression studies. So with that, we start with the procedure of RNA-seq. By now, seeing the first slide itself, you might have understood that there are basically 4 major steps. Okay, the 4 major steps are RNA isolation, cDNA library preparation, sequencing, and data analysis. Today's session will be covering the first 2 steps. And under these steps itself, I'll be covering many other small topics, and I really hope it will be useful for you. Okay, so the first step, RNA isolation and selection. While working with RNA, the things that we need to keep in mind is RNA is very labile. Okay, it's not as stable as DNA. So it's a bit difficult to work with RNA. So they're just easily degraded. Also, RNases are way too stable than other proteases. So the combination of both of this makes it very difficult to actually get a pure isolated mRNA at hand. So depth of RNA isolation, total RNA isolation has to be done very carefully. So please do keep that in mind. Also, to avoid DNA contamination, you can add DNases to remove any traces of DNA present in the sample. Okay. Now, the basic of RNA isolation is that first you homogenize a sample, homogenizes breaking down the cell wall, and then you carry out liquid-liquid extraction or solid phase extraction. If you have any doubt, please do refer to it. There will be a lot of data available on the techniques, how RNA is actually isolated. Okay. So let's see what happens after RNA isolation. Okay. So throughout the procedure of RNA-seq, right, one very important thing that we need to do is quality check at every stage. Okay, we need to check the quality of the total RNA that has been isolated. After library preparation step, we have to see the quality of the library being prepared. After sequencing step, we have to see the quality check the reads that have been, the sequences that we have. And during the data analysis, also a lot of processing has to be done. So at every step, there is kind of a quality check that is happening. That's a very important part of RNA-seq. Okay, so under that itself, let's see what is something called RNA Integrity Number. Okay, so that is kind of a parameter that can help us to understand the quality of the total RNA that you have just isolated. Okay, so traditionally RNA integrity was assessed via gel electrophoresis, that is by visual inspection of the ribosomal RNA bands. So you know that total RNA will have rRNA, and when you analyze the gel, in case you get two intact bands corresponding to 28 and 18 as rRNA, that means your total RNA of its integrity is retained. Okay. One second. Okay. So the 28S rRNA band is approximately twice as intense as the 18S rRNA band. So it's a ratio of 2:1. Okay. As the RNA degrades, the 2:1 ratio of high quality RNA decreases and low molecular weight RNA begins to accumulate. Okay, so here you can see this is intact total RNA. You can see clear two bands over here. This is partially degraded total RNA. You can see the bands are, kind of spread across the whole place. This is heavily degraded total RNA. So human interpretation of gel images is subjective and inconsistent. So initially we were just visually understanding, right, whether it is degraded or not, looking at it, right? So to minimize the errors in that, we developed a software algorithm that allows for the calculation of an RNA integrity number, that is RIN, okay? From a digital representation of the size distribution of RNA molecules. So this is kind of how the digital representation will look like. The RIN number is based on a numbering system from 1 to 10, okay, with 1 being the most degraded and 10 being the most intact. So all the samples are actually assigned a particular number from 1 to 10, depending on how intact your RNA is. If it is very intact or not degraded at all, the RIN will be 10. As simple as that. Okay, so this approach provides a means by which samples can be compared in a standardized manner. So you can see over here an example of RIN when it is 10, right? You can see two clear peaks over here. When RIN is 6, you can see different bands or different peaks over here distributed all across the graph, right? And RIN 2. So the peaks are not at all there for 18S and 28S. The peaks are not at all there. So that is giving you a number of RIN 2. Okay, so that is how you actually understand the quality. And there are also kits available wherein you can actually repair your degraded RNA and then go forward to the next step. Okay, so quality check is important. After that, once you have your isolated total RNA, let's see what has to be done. Okay, now when we say total RNA, about 85% of that sample will be composed of rRNA or ribosomal RNA, and about 10 to 12% might be your tRNA, and some 2 to 3% might be mRNA, and a very less percentage might be your long non-coding RNA, small non-coding RNA, circular RNA, and microRNA. Okay, so this is the basic composition of your total RNA that you have isolated from your sample. Okay, out of that, we will be focusing on mRNA. Okay, because mRNA is the one which almost kind of directly gives us an indication that how much a gene has expressed. Because after transcription, you get mRNA. mRNA is what is being translated into proteins. So our focus will be to actually isolate this mRNA out of this total sample, RNA sample. Okay. Now, depending on your studies, you might want to work on the other types of RNAs also. But since we are focusing on gene expression and gene expression studies, we will be focusing to get mRNA out of this total samples. Now, to isolate this particular mRNA species from your total RNA, you have two approaches, two major approaches. Okay. So the first approach is using poly-T oligos. You might all be aware that mRNA, after post-transcriptional modification, it has a poly A tail at the end, right? So what we do is we actually use magnetic beads that have poly DT attached to them. Okay, that's thymine attached to them. And we'll selectively target poly A tail mRNA. So for example, in this diagram, you can see this is your mRNA, and suppose this is your normal other RNA. This is your bead that is coated or attached with oligo DTs. Okay, you can see over here now. So what will happen, since adenine is complementary to thymine, only the mRNAs with those poly A tail will go and attach to these beads, and they will be pulled out from the, when you take out the beads, right, the mRNA will be separated from the pool. So that is one approach. Another approach that you can use is rRNA depletion. That means you have certain kits available, for example, ribo minus or ribo 0. What they do is they will specifically go and degrade or deplete rRNA. So since rRNA is the major chunk, that will be depleted. And you'll be left with other types of RNA. And this is especially useful when we want to study those small non-coding RNAs or long non-coding RNAs, or even work with micro RNAs and circular RNAs. If you want to work with those, this method is much more preferred. Why? Because in the first step, only mRNA will be enriched, right? But in the second step, or in the second method, since only rRNA is removed, you will be left with all the other types of RNAs also. So you can work on micro RNAs also using this technique. Okay. So that was the first step, that is RNA isolation and enrichment, wherein you learned about the quality check also. Okay. The second step, or one of the major steps in RNA-seq, is the library preparation. Okay, so let's see what is library preparation. So after your RNA is isolated, the next step is to prepare the RNA-seq library. Now, what protocol you follow for preparing this library will depend on which RNA species you are selecting. Like, for example, is it mRNA you're working with, or are you working with microRNA? So depending on the species, it will also depend on which sequencing platform you're using. There are different types of next generation sequencers available. You have Illumina sequencers, you have Ion Torrent, you have solid sequencing, pyro sequencing, different types. So which sequencing platform you're selecting and which RNA species you're selecting, based on that, you will have a protocol for the library preparation. Now, in today's session, that is this particular session on RNA-seq, we'll be focusing on Illumina sequencing, that is sequencing by synthesis method by Illumina. So our library preparation step will focus on adding those kinds of adapter which are compatible with the Illumina platform for sequencing. Okay. Now, what is a library? So in high throughput sequencing terms, a library is nothing but a random collection of DNA fragments that are ready for sequencing with a specific protocol. That's it. You're just making your sample ready to be sequenced. That is what is happening in library preparation. Basically, library preparation is the second major step, but under this also there are many mini steps. Okay, so the first step will be RNA fragmentation. Second step will be reverse transcription. Third step will be adapter ligation. Fourth step will be amplification. Okay, so this is all under library preparation. These 4 steps are under library preparation, which is the second step of RNA-seq. So let us see the first step, RNA fragmentation. Why do we need to do RNA fragmentation? So you have isolated your mRNA. Now, whichever sequencing platform you are going to select, they will have a specified length that we have to provide. Okay, fragment length should be only so and so. So in order that the sequencing happens in the right manner, we need to prepare our RNA of a particular length. So for that, you'll have to carry out fragmentation. Now, And so that is what is being said over here. mRNAs are typically fragmented to smaller pieces of RNA to enable sequencing. Okay. The next step is reverse transcription. Okay. So I hope by now you know the concept of reverse transcription, right? Transcription is DNA to RNA. So reverse transcription is when you go the reverse way, RNA to DNA, back to DNA. It is done in the presence of an enzyme called reverse transcriptase, which is specifically isolated from retroviruses, because viruses have viral RNA. Okay, they have their genetic material as RNA, and to get into a host, they actually need to reverse transcribe the RNA to DNA so that they can integrate themselves to the host, and when the host replicates, the virus also replicates itself. So basically, that is why they have it. We use these reverse transcriptases to carry out our reverse transcription reaction. Okay, so you have your mRNA. The first strand of cDNA will be synthesized. Now, this first strand of cDNA is synthesized maybe using oligo-DT primers or using random hexamers. So why do we use oligo-DT? Basically because we are lucky because mRNA has a poly-A tail already. Okay, so if you use a DT primer, oligo-DT means many number of T's, okay, thymine joined together. So if you use that as primer, that primer can come and exactly attach itself over here, okay, complementary to the adenine. Here I've shown 3 A's. It does not mean it has only 3 adenine at this end. It's just a representation of a poly-A tail, okay. So your primer will come and attach over here. Reverse transcriptase enzyme then can start forming the first strand of cDNA. So whatever is the base that is complementary to this will come and attach over here, and so on and so forth. That will take place. So what you get as a result of this is mRNA DNA hybrid, right? mRNA DNA hybrid. Also, we need to remember, not only do we use oligo-DT primers, we also use random hexamers. Hexamers will contain 6 nucleotides. Okay, so we randomly design certain nucleotides of 6 base pairs length, base length, so that they will randomly go and attach wherever they find the complementary part, and from there they will extend. So random primers are also used. Random hexamers are also used for the first strand synthesis. Okay. Then comes the second strand synthesis. So we need cDNA as a double-stranded molecule. So we need to create the second strand of cDNA. Now there are a lot of approaches by using which we can create the second strand of cDNA. The commonly used ones I have put over here, but it is not limited to this. Okay, so this is just a simple diagram to show the 3 common approaches that can be used to make the second strand of cDNA. Okay. So the first method could be, so remember I'm talking about this, okay, this is the beginning. You have your mRNA DNA hybrid. On this, we'll be doing the next steps. So one method could be, you could add NaOH. NaOH will degrade that RNA strand, mRNA strand, and you'll be left with only that single strand of cDNA, first strand. This cDNA, generally the 3' end has an ability to form a loop. Okay, it kind of forms a hairpin loop. When it forms a hairpin loop, this particular portion over here, it kind of acts like a primer, right? It kind of acts like a starting point. Then what will happen if you add DNA polymerase and nucleotides? This will be synthesized. The second strand will be synthesized. And after that, since there's a loop over here, you need to use S1 nuclease to cut this loop, and you have your double-stranded cDNA. That is one approach. Okay. Another approach could be the use of RNA-seq. Now, RNA-seq, as the name suggests, it is going to cut RNA, right? RNAs will cut RNA. So what RNA-seq does is, it goes and particularly creates nicks or breaks in this mRNA strand. Okay, it removes nucleotides from the mRNA strand. After that, during that process itself, if you're going to have DNA polymerase 1, with the help of DNTPs, this DNA polymerase 1 goes and fills up wherever the gap is there. Again, this process is repeated. I mean, this happens maybe simultaneously, you know, like RNA-seq goes and cuts, forms nicks, and simultaneously you have DNTPs being filled in by DNA polymerase 1. Finally, what you will have is a complete double-stranded cDNA. Okay, the second strand is synthesized like that. Another approach is the use of terminal transferase to add DGTPs to one end. So first of all, you'll be adding NaOH, that is to denature the mRNA strand, that will be removed. After that, terminal transferase will add Gs to the 3' end of the cDNA. Okay. Once you have Gs at one end, you can use oligo DC primers. C is complementary to G, so you will use oligo DC primers. Plus, in the presence of DNA polymerase and nucleotides, what will happen? The primer will come and attach, and the synthesis of the second strand will be done. So finally, you will have a double-stranded cDNA. So you can go with any approach to get your double-stranded cDNA. Basically, we are not going to work with mRNA directly. We are going to focus on cDNA. Why could that be? One very simple way to explain it is that RNA is not that stable. It's not easy working with RNA as it is easy to work with DNA. So if you have a DNA molecule, it's much more stable and sequencing part is much more easier. So with that, we come to the end of today's session. In this part, we learned about RNA isolation and selection, and we just started with the library preparation method, under which we studied RNA fragmentation and the next step of reverse transcription to get us cDNA. If you found this video useful, please do let me know in the comment section. Please do not forget to subscribe to my channel. It will be a great help for me. And I will meet you with the part 2 of this video very soon. Until then, bye-bye.